We will move on to the final speaker of today, a veteran of the Aging Research and Drug Discovery meeting. I'm not sure if you've been to all of them, maybe close to. Vadim Gladyshev.
Thank you, Morten. And thank you very much for the invitation and chance to speak at this really outstanding conference. Okay, great. Thank you. So what is aging? Is it age-related changes, increased mortality rate, decreased fitness, or loss of function, or continuation of development, increased biological age, or damage accumulation? So in the second half of our life, all of these features apply to the aging process. However, there must be one most important feature fundamental to the nature of aging, which would define or result in a consequence of other features that represent aging. So it is very important to understand what this fundamental feature is, because we need to know our enemy. We need to develop interventions against that particular feature, instead of all of these features which are listed here. And I'm a little bit surprised that relatively few people experimentally test this in the lab. But hopefully, after listening to this talk, some of you will join us, whether you as a lab member or collaborator or funder or investor. So to begin to address these questions, we developed a research program to quantify longevity, aging, and rejuvenation, which would lead to the identification of anti-aging interventions. And for this, we approach this from three different angles. One is longevity. Here we test for the potential to live long. So we study longevity across cell types, longevity across species of mammals, and also longevity in response to diverse interventions that extend lifespan. This results based on omics approaches in so-called longevity signatures. We also try to quantify aging, where we develop epigenetic clocks, also clocks that work at a single cell level, and also clocks that are very inexpensive, because currently it's a problem of the cost. And second or third, we also try to develop omics approaches to rejuvenation, because the trajectory of aging is not necessarily the same as the trajectory of rejuvenation. So from these three angles, we then approach aging and rejuvenation by trying to quantify it and then develop approaches and candidate compounds which we then test in animal models and hopefully in the future in humans. So just give you a few examples in terms of longevity signatures. We published over the years many studies on the genomes of long-lived species. Some of them are shown here. Just two studies recently, just to highlight the recent developments. It's a beaver and neck rat chromosome level assembly, as well as another paper on 200 mammalian species. We also do this across mammals. Published studies at the level of the transcriptomes, metabolomes, ionomes, and currently expanding even further in that direction. So this is the example of longevity signatures across interventions, where we analyzed 17 different ways that extend lifespan of mice, ask a question whether they are related to one another, whether they modify the animal in a similar way. Apparently, there are many routes to longevity, so they change mice. Actually, some of them act in a similar way, like, for example, shown here in red, many interventions, but many of them work in different ways and still extend lifespan. These signatures can be used to identify new interventions. Here on the upper part of the slide, we tested public data. Basically, somebody sequences a particular condition, loads in this database, and we extract and analyze. And on the lower part, we screened drugs, and some of the 4 drugs, which are shown here, we identify as candidates for lifespan extension. We're currently testing these and many other that we identified. So we have a large program on that. So, to understand also aging and quantify it and test interventions, we need robust biomarkers. I think there has been a revolution in the field, more or less a revolution started by Steve Horvath, who developed the first robust clock to quantify biological age. And of course, now many clocks are available, multi-species, multi-tissue, tissue-specific, and clocks trained on chronological age or phenotypic age or future mortality and so on. Also, the same thing in mice. In mice, quite a few clocks have been developed, those in blue, which come from our lab. And of course, there are many other clocks, not only epigenetic, but also clocks that work on different, quantify different parameters. They also work. So this is just an example of our clock. So you can see on the left, we tested the effect of calorie restriction. First, in blue are control. This is 16 age groups, and you can see a good correspondence between the actual age of the mice and predicted methylation age. 4 groups in red. These are calorically restricted mice. And you could see that at any chronological age, clocks tell that these mice are biologically younger than their chronological age. In the middle panel, growth hormone receptor knockout mice, the knockout mice are, again, younger than the age-matched, chronological age-matched controls. And on the right, we collected fibroblasts from kidney and lung of animals, converted them to iPS cells, and actually the predicted age of the iPS cells is close to 0. So one feature I wanted to say of this clock is that they actually detect lower ages. So for example, on the left, you could see like 2 blue circles, small circles in the younger ages. These ages were not part of the model, but the age could still be predicted. And of course, on the right, the age of iPS cells also could be predicted, even though the clocks, in this case, were developed for blood in the adult ages. However, current clocks, epigenetic clocks, and actually many other clocks, they have 3 problems. So first, they are expensive. So to the order of $200–300 or maybe more per sample. It's not feasible, for example, to test various, let's say, genetic screens or large clinical studies. Also, they only quantify aging in bulk tissue, whereas aging happens at the level of individual cells. And the third, they are trained on age-related changes, all of the age-related changes, whereas what we need is to train them on the nature of aging, what I think is damage, basically. Quantify damage that accumulates, not just any related changes. For example, if you train a clock based on the use of like a glasses or hearing aid, they would quantify the aging process, but they don't represent aging, right? They present adaptation that we use to the aging processes. In the same way, many other changes, it's a combination of damaging, neutral kind of changes and adaptive changes. And the clocks, they all quantify this, whereas what we need is the clock that tracks really aging. And that clock does not exist yet. So what I want to tell you is that we developed a new system called a flexible and scalable probabilistic framework for epigenetic age prediction. And Alex Trapp, who sits right there, so he will give you, will present this in more detail on September 3rd. He will tell you 2 things: single-cell epigenetic clock, and also a clock that works at a very low cost, at a level of 100 to 1,000-fold reduction in sequencing costs. Just to highlight that, this is how a single cell that he developed applies to individual mouse hepatocytes and embryonic fibroblasts. On the left, you can see mouse embryonic fibroblast. Each dot is 1 cell. So we predict the age of each cell by sequencing from about 10,000 sequence reads. In the middle, it's hepatocytes from a 4-month-old mouse. And you can see dashed blue line, which is the 4 months, and you can see that the individual cells, they correspond approximately to that. And on the right, these are individual hepatocytes from a mouse, which is 26-month-old. So from these studies, what it seems like, even though most of cells, they age according to their chronological age, there are some cells that age faster and some cells which age slower. And if you look on the right, that's how the tissue actually looks like. There's a lot of heterogeneity in the aging of individual cells, and we need to be able to quantify this in the future studies if we want to study aging. So now I want to tell you a little bit about rejuvenation, because so far I told you about the aging process, which is vertical direction in this slide. And aging here is represented by water. In these steps, we kind of slowing down the aging process. We can do it by calorie restriction or rapamycin or whatever other interventions. But what we really want, in addition to slowing down aging, we want to take an organism from the older state and pump it to the younger state to really rejuvenate it. And of course, this is possible, and we know it's possible, from the example of the conversion of fibroblasts to iPS cells, which I already presented to you, which is like Yamanaka-like reprogramming, of cells. To give you a second example of rejuvenation, let me first kind of discuss these basic ideas of August Weismann from the 19th century, who proposed the separation of the ageless germline and aging soma. You can find thousands of papers which literally say this: germline does not age. And of course, it should not age, otherwise the next generation would start from an older age. Yeah, we all start aging from the same young age, so regardless of the generation. However, germline, it's a live system. It's a metabolically active cell. It must accumulate byproducts of metabolism, or mutations, epimutations, modified proteins, and so on, which means that the germline does age. For new life to begin in the same young state, biological age must somehow be decreased somewhere from the age that's accumulated in the germline. So we have a hypothesis that the germline ages during development and adult life and is rejuvenated in the offspring after conception. So basically the model is like this. The life begins at the zygote level stage with the age which is not zero. And then it decreases to the state which we call ground zero, which represents the lowest biological age of the organism. And then it increases. So this is a hypothesis. So one prediction from this hypothesis would be that if we take cells somewhere in between zygote and ground zero state, these cells should not age. So we tested this by studying embryonic stem cells and iPS cells. So here are the cells which were passaged up to 100 or even more times, embryonic stem cells, and you can see that there is no aging of these cells. If you take fibroblasts in culture, many other cells, they would age, right? But embryonic stem cells do not age. And also iPS cells, you can see on the right, they were passaged 40 times. Again, you can barely see any aging. So the age of this is consistently below zero. So, and then we directly tested this hypothesis by actually studying the embryos and applying clocks, different clocks. Just one clock is shown here, but actually we applied various clocks developed in my lab and also in other labs, and we consistently see that the biological age decreases during early development. We have not defined the actual ground zero state. It's somewhere approximately in mice embryonic day 7. But again, so we cannot be sure exactly, but we clearly see it's reduced. And then it increases after that, approximately at the stage of gastrulation. On the right, you can see application of a single cell clock, which is a completely different approach. We have this probabilistic approach versus kind of machine learning elastic net approach. It's completely different, and it also shows the same biological age reduction in mice between the embryonic days 4.5 and 7.5. In each cell, again, individual cell here, the age is predicted for those cells. So this is what it appears, that when life begins at the zygote state, age is not zero. And then at some point, age is decreased to a certain level, embryonic ground zero, and then aging begins. This ground zero marks the beginning of biological age of the organism, based on this data. So in a broader sense, what it tells, I think it tells a little bit about the nature of aging. So in my mind, aging is accumulation of damage that increases biological age. So it's not increased mortality and not loss of function and not other features that I told you at the beginning of my talk. Aging begins at ground zero and not at the completion of development like most evolutionary models would predict. And it ends, of course, when organism dies, and it has no purpose because the aging is not programmed. On the other hand, development has a purpose. The purpose is to develop a fit organism. Development and aging, they go in parallel, hand in hand. And aging is basically a consequence of development in early life, and not continuation of development. It begins at conception and ends in humans around the age 20. Then rejuvenation would be a damage clearance or reduction that decreases the biological age. And again, so the trajectory of aging and trajectory of rejuvenation might not be the same, probably not the same. And again, so I tell you that we identified first natural rejuvenation event that begins at conception and ends at ground zero. So we don't know yet what this means. We don't know in which particular cells it happens, but we want to identify this at individual cells and understand exactly how it happens. So it's actually a very exciting direction. And what about immortal germline and aging soma? Of course, the Weizmann ideas are great ideas. However, now it's clear that both soma and germline can age, and they can be rejuvenated. And if we identify how they can be rejuvenated, maybe we could try to induce this process in adult cells, in adult organisms, of course, sometime in the future. So finally, I'd like to acknowledge people who have been involved in this work. On the left, on the upper part, is the current lab members. Many of them contributed, but I would particularly point a person second from the right. It's Alex, again, Trapp, who developed a single-cell clock, and a person second on the left, Csaba Kiripeshi, who provided evidence for this embryonic rejuvenation. Other people have been involved. I also mentioned data from Alex Tyszkowski and Daniel Petkovic in my talk, as well as you can see many collaborators. We collaborate on aging and longevity and other features. And all of this work is funded by National Institute on Aging. So thank you very much.
Thank you so much. Thank you, Vadim. That was a really fascinating talk. I can maybe start with a question. So have you applied your single-cell age predictor on the gastrulated embryo to figure out which population is...
Wait for Alex's talk.
Okay, that sounds fantastic. Do we have any other questions from... We have Vera Gorbanova.
Vadim, very stimulating talk. So do you have thoughts of what exactly is being repaired between conception and day 7? Like what type of damage? And would there be certain repair systems that are really working hard within those 7 days?
Well, presumably all of the damage should be repaired at this point, right? Somehow it is repaired. We know that telomeres are extended starting from zygote and later on. Yeah, we know that methylation is initially, is kind of cleared, removed, and then remethylated. And actually what we find is that the lowest biological age does not correspond to the lowest methylation. It corresponds to the highest methylation. So methylation is first stripped and then remethylated and corresponds to the highest biological age. So apparently what cannot be rejuvenated is the mutations which appear during in the germline in the previous generation. Yeah? However, we think that rejuvenation of the genome happens at the species level. So, for example, certain combinations of mutations in the offspring, which are particularly damaging or deleterious, they will be removed by negative selection because this organism would not be viable, probably, and that's probably one of the reasons for kind of early life mortality, like during pregnancy, for example. Again, so this is potential ideas for future research.
Very nice. We have a question on Slack here from Dmitry Kryukov. Can the rejuvenation of germline during embryogenesis be just a consequence of extracellular matrix reset?
Well, it's unlikely to be just extracellular matrix because, again, so damage accumulates in cells at multiple levels. And if the damage persists there, then the next generation would start in a little bit older age, and we don't see that. So it must be somehow completely cleared. I'm not saying that this is the only process of rejuvenation. Of course, it's also possible that a partial rejuvenation happens at the level of development of the germline. And Cynthia Kenyon, actually, she has some great studies in support of that idea as well. Perhaps it's distributed also across different cell types. It does not necessarily mean it's like all happens exactly at the same time. We say that at the level of the entire embryo, we see a minimum, but the rejuvenation might happen in different cell types, in a slightly different stage, but we don't know that yet.
There's the epiblast population grows a lot at that moment and then invaginates. And is there a possibility that the cells that actually proliferate are the ones that are most healthy?
It is possible, yes. Needs to be studied.
Interesting. Shall we do maybe one more from? Do we have more from the audience? We have. There's a question about the technical aspects. That there's one model used, elastic net, which lacks homoscedasticity at the ends of intervals of prediction. How this results of minimum of epigenetic age of day 6 of development can be validated. I guess it's about question about the clock model.
Yeah, so apparently any clock that we apply, they actually shows the same. Well, of course, if one clock would show this and other clocks would not, it would be hard to say because we don't know which clock needs to be used.
Yeah.
So we used our clocks. So Petkovich clock, for example, we used a clock from Wolf Reich's lab, Stubbs clock from Steve Horvath, and Alex's single-cell epigenetic clock, which are all completely different. They all show the same.
Very interesting.
But again, of course, needs to be validated in other labs as well. So.
Very good. I think we'll stop here and thank Vadim for a fantastic talk.